This is part 2 of our comparison between skeletal muscle and cardiac muscle structure and function. So far, we've talked about action potentials and excitation contraction coupling. Next, we'll talk about the differences between fiber structure. We'll discuss motor units and how that affects contraction strength, and we'll finish up with talking about the control of muscle contraction, either by nerves or by the automaticity of the cardiac muscle itself. A quick review of the major difference between cardiac muscle action potential and skeletal muscle action potential. The cardiac muscle has this extra phase, this plateau phase, caused by the opening of voltage-sensitive calcium channels. This makes the cardiac muscle action potential way longer than the skeletal muscle action potential. We have this extra plateau phase increasing the length of the action potential. This allows you time for proper blood ejection from the ventricles. Next, let's review the structure of skeletal muscle cells and compare that to the structure of cardiac muscle cells. First, let's talk about skeletal muscle cell formation. Skeletal muscle fibers start out as myoblasts, or precursor cell. During development, myoblasts will fuse, creating these very long muscle cells. Please note, muscle fiber and that word muscle cell are the same thing. One muscle fiber is a very long muscle cell. Skeletal muscle fibers, because they're so long as they resulted from lots of few cells, have multiple nuclei. We can now look how each muscle fiber is arranged in an entire skeletal muscle. Each muscle fiber is surrounded by a thin membrane called an endomysium. This has the very important function of electrically isolating each skeletal muscle fiber. That means that each muscle fiber, if it's activated and contracting, the entire length of the muscle fiber will contract, but that action potential will not jump from one skeletal muscle fiber to its neighbor. Each skeletal muscle fiber has to be told to contract by a motor neuron. We'll come back to that. It's a very important point. Muscle fibers are arranged in groups called fascicles, again surrounded by a membrane, and each fascicle is going to run the entire length of the muscle. At the end of the muscle, each muscle fiber is hooked up to a tendon, which is going to be some dense, regular connective tissue. Tendons form the attachment between muscles and bones. And a review of the structure of each individual skeletal muscle fiber. We do have an endomysium and a sarcolemma wrapped around it. Loaded in here are going to be the contraction machinery itself. We have myofibrils, which are collections of our myofilaments, the thick and thin filaments. Each group is arranged into a sarcomere. This gives the muscle its striated or the stripy appearance. The two other pieces here, we have our T-tubules, which take the action potential from the outside sarcolemma deep into each cell. And then we have our sarcoplasmic reticulum. And this will be the area where we store our calcium, which gets released after an action potential comes down the T-tubule. How does our cardiac muscle cell compare to the structure of the skeletal muscle units? First, the easy parts. Our myofibrils and our sarcomeres, the structure is about the same. They're a little less organized, but cardiac muscle cells are also going to be striated. The contraction machinery works about the same as skeletal muscles. We have T-tubules, there is sarcoplasmic reticulum, but there's just less of it. We don't need to have as many of these as we can get calcium going into the cell from the outside of the cell through those channels. Next, each individual cardiac muscle cell is nowhere near as long as a skeletal muscle cell. Cardiac muscle cells are fusions of those precursor myoblast cells, but they're usually only a couple of cells that are fused together. So cardiac muscle cells will typically only have 2 nuclei versus lots and lots like cardiac muscle cells. Next, cardiac muscle cells are much shorter and stubbier than skeletal muscle cells. Each muscle cell is going to be joined up by these special joints called intercalated discs. More on those in just a second. The other major difference is cardiac muscle cells are usually branched. They are not long single tubes like those skeletal muscle fibers. What do these guys actually pull against? What they don't pull against is tendons. They actually pull mostly on each other. Those intercalated discs create very strong junctions between these cells. What about tendons? There isn't really a distinct tendon in a heart, but there is a lot of connective tissue. It's covered in the other part of the lecture. It's called a fibrous skeleton of the heart. The fascicles of cardiac muscle tend to be wrapped around the heart, but not just simply wrapped around like a circle. They're actually arranged in a complicated pattern that goes around but also twists around the heart like this. When the cardiac muscle contracts, it's just like squeezing the heart, but more like wringing out a wet, damp rag or a sponge. It's a squeezing and then a twisting motion. This results in a very efficient ejection from the ventricles. Let's go back to those intercalated discs, those joins between our stubby cardiac muscle cells. These intercalated discs contain two types of cell-to-cell junctions, which we learned about all the way back in chapter 3 or 4. We have two major ones here. One is not shown. We have lots of desmosomes in intercalated discs. Those are the cell-to-cell junctions that create very strong joints. It's obvious these guys will pull on each other. They need to be stuck together very well. The other type of cell-to-cell junction, they show them down here, and these are our gap junctions. They serve another very important function. Remember, back in skeletal muscle cells, if one skeletal muscle fiber is activated, that activation will not cross over to its neighboring cell. Cardiac muscle cells, we have a very different function. If we activate just one cardiac muscle cells, the depolarization in this cell will easily jump across from this cell to the other. When we depolarize, remember, sodium will flood into a cell from those open channels. Sodium can easily diffuse from this cell over here. The end result, if this one cardiac muscle cell depolarizes, all of its neighbors will depolarize. So we only need one cardiac muscle cell to depolarize, then it will depolarize every single cardiac muscle cell in the heart. This is very, very different from skeletal muscle. So far we've established that skeletal muscle fibers are electrically insulated. Each fiber is not hooked up to the neighboring one. In cardiac muscle, it's the opposite. Each cardiac muscle fiber is electrically hooked up to its neighbor. The question is why? Let's go back and review the activation of skeletal muscle. First, skeletal muscle will not contract unless it's told to by a motor neuron. A quick review of how that works. At the top here we see a motor neuron coming down and splitting off into several end plates. If we look at the end plate here, this area here is going to be called a synaptic knob. A brief review of the events. Action potential travels down the axon. Once the action potential, the depolarization, gets down to the end of the synaptic knob, we will open up a set of voltage-gated calcium channels. Calcium floods into the cell and sticks to synaptic vessels. Once calcium sticks to these vessels, the vessels will release their contents, neurotransmitter, into this gap here called the synaptic cleft. Neurotransmitter diffuses across and binds to these receptors on the muscle sarcolemma. These receptors are actually receptors and channels. Once the neurotransmitter binds to the receptor, these guys open up, sodium floods into the cell. This is that initial depolarization that'll set the neighboring sodium channels on either side here over that threshold value. And this will start the action potential over here in the muscle cell. So once again, this means that skeletal muscle fibers will only contract if they're told to by a motor neuron. This motor neuron here, you can see it splits into several ends. Each of these muscle fibers controlled by one individual neuron. You'll see motor neuron 1 here controlling these red-colored muscle fibers. This will be called a motor unit. A motor unit is defined as one motor neuron plus all of the muscle fibers it activates. If this motor unit is activated, will another motor unit next to it be activated? Only if told to by the other motor neuron controlling these. Why do we have multiple motor units in skeletal muscle? This is how we control the strength of contraction. Over here we have skeletal muscle tension. Down here we have the number of motor units activated. If we just need to have a small weak contraction, we can activate just 1 or 2 motor units. If we want a muscle to have a stronger contraction, we can add motor units until we max out our muscle. The term here is called recruitment, and this is how skeletal muscles control the strength of contraction. Do we need separate motor units in cardiac muscle? Is there ever going to be a time where we only want to activate a certain number of cardiac muscle fibers? And the answer is an obvious no. Every time your heart contracts, you want to activate every single fiber. You can think of the entire heart as being a single motor unit. We will cover how we adjust the strength of contraction, but it's nothing to do with recruitment. There is a mechanism to just tell muscle fibers to contract with more strength. That has everything to do with the sympathetic nervous system, and we'll get to that eventually. So how do we initiate an action potential in cardiac muscle cells? Just to be clear, we do not do this with a motor neuron. There are no motor neurons going to the heart that start an action potential. There are neurons that go and innervate the heart, but these have everything to do with the sympathetic and parasympathetic nervous system. They will not initiate action potentials, but they will adjust the rate of the heart rate and the strength of contraction. So to start a cardiac muscle contraction or action potential, we need to figure out how to do this. There turns out to be a very limited number of special cardiac muscle cells which function different from the rest. Only about 1% of cardiac muscle cells have this special function. And these cells, they have several names, but they're often called nodal cells. The secret here is to understand that we have a different set of our voltage-gated channels. Remember, in your typical cardiac muscle cell, you start the depolarization with fast voltage-gated sodium channels. You maintain the plateau phase with those voltage-gated calcium channels, and you will reset it, you will repolarize with our very slow voltage-gated potassium channels. In nodal cells, all of these channels are slightly different, but the main difference is in the sodium channel. Remember, we called our potassium channel in both the skeletal and our regular cardiac muscle cell a fast voltage-gated sodium channel. We now have a new one, and this is going to be our slow voltage-gated channel. And this sodium channel functions differently from just about every other cell. We will also have our same fast voltage-gated calcium channel and a slightly different voltage-gated potassium channel. So keeping our different channels in mind, let's examine this special action potential found in our nodal cells. We'll start here at our resting membrane potential. We've got approximately 60 millivolts here, but it's close enough to 70. The secret here is these special nodal cells, they do not remain at resting membrane potential. Once they get down to resting membrane potential, this actually opens up our brand new slow sodium gated channels. So at this point right here, our slow sodium gated channels will open up. They're slow because they don't let much sodium in at once. This results in this steady depolarization. Once we depolarize above our threshold here, then our calcium, our fast voltage-gated calcium channels will open. This is very different from every other cell we've seen. This depolarization phase here is not due to fast sodium-gated channels. They are not found on these nodal cells. It's our voltage-gated calcium channels that do our depolarization. Once we've depolarized, we don't have much of a plateau phase. We are going to start to repolarize here. Our potassium channels will open up, giving us a steady depolarization back down to our resting membrane potential. The entire process takes approximately about half a second. Well, this is showing about 0.8 of a second. Once we get back down to resting membrane potential, we repeat the entire process. Our slow voltage-gated sodium channels open, We will have our depolarization phase due to our calcium channels, and we will repolarize. The end result is these special nodal cells, they will never sit at resting membrane potential. They will automatically start this slow depolarization up to threshold. This slow depolarization is called the pacemaker potential. And this is where that term pacemaker comes from. These cells set our steady rhythm of depolarization and action potentials for the heart. Once these cells depolarize, the neighbors will depolarize because of gap junctions, and your wave of depolarization or action potential will spread from this point around the heart. Those special nodal cells, they are not spread evenly around the heart. They're really only found in two areas, and these areas are called our nodes. Most of these nodal cells will be found all the way at the top of the atria, in what's called our sinoatrial or SA node. There are another group found down in the second node, which is our atrioventricular node, found at the junction between the atria and the ventricles. This node up here is also called our pacemaker. This is where most of the nodal cells will be found. This is where we will start each individual heartbeat. The action potentials will start here and spread from the top of the heart down through the rest of the heart. The heart has what's called a conduction system, which transports the action potentials to various parts of the heart in a very special sequence of events. But that we'll cover in a separate lecture.